Bismillahirrahmanirrahim, students. Hope you all are fine. Assalamu alaikum wa rahmatullahi wa barakatuh. Well, we are going to work on the DNA replication. That what is the DNA replication? Basically, if we are going to see the DNA replication, that it means that how the DNA used to replicate. Let's see that, as I told you before, that the DNA is made up of two double-strand helical structure. So it means that it contains the phosphate group, it contains the ribose, as well as it contains the nucleotide. If you remember, like adenine, thymine, cytosine, guanine, cytosine, thymine, adenine, thymine. So these are basically these nucleotides. These are very much important. Fine. So in this backbone, this strand is made up of ribose and the deoxyribose sugar, as well as the phosphate. So what is happening, student, and how the DNA replication used to occur? It is a very much interesting topic. First, these two strands, you know that the hydrogen bonds used to exist in between them. They are the hydrogen bonds that used to exist in between them. So it is like that. First, these hydrogen bonds are going to break. They are going to unzip themselves, like these bonds are going to get break, obviously by the help of the enzyme, and it is just like that to unzip the bag, like to unzip something. Like जैसे आप zip बंद करते हो या खोलते हो, when you are going to unzip the zip, what will happen? It used to separate into two strands. So similarly, when these bonds are going to break, these two are going to separate into two strands. So it would be like that. One strand would be here and one strand would be here. Okay? So that is like A, C, G, T, A, and this is another strand having the T, G, C, A, T, and these both strands are going to act as a template. Now what will happen, beta? The new nucleotides will come and join with these nucleotides and will form the new strand. And now what will happen? This one strand is now going to have been converted itself into two strands. Like the nucleotides from the blood or from the system body, from the cell, will come not from the blood, students. It's like from the cell. So the nucleotides from the cell will start joining in front of it. Like adenine, obviously the thymine will come. Cytosine, guanine, cytosine, thymine, adenine, thymine, and will make the bond with each other. Similarly, in that case, adenine with thymine, double bond. Cytosine with the guanine with the cytosine, thymine with the adenine, and the adenine with the thymine. So you can see that these both strands are just a ditto copy of this strand. So this is known as the replication of the DNA. So if you are going to read this paragraph from your book, let me tell you, it is written here clearly, if you are going to read that we have studied in grade 9 cell cycle that before a cell division the DNA is replicated. हम ये पढ़ चुके हैं. But the question is that how it's replicated. It is a very much simple question. That answer is very much simple. It is done to make the copies of the chromatin of the chromosome. During replication, the DNA double helix unwound and two strands are separate, much like the two sides of the zipper. The way I have told you, beta. Each strand acts as a template to produce another strand. What is the meaning of template? Means that this strand is going to help to produce the new strand. So that is, it is called as a template. Its nitrogenous bases make pairs with the nitrogenous bases of the new nucleotides, and in this way, four template strand will make a new polynucleotide strand. Polynucleotide means having a lot of the nucleotides. So each template and its new strand together then form a new DNA double helix that is going to be identical to the original.